Good morning and welcome. I'd like to thank everyone who has taken time out of their busy schedules, both today and over the past month, for the Let's Get Real webinar series on patient registries and real-world evidence. I'd especially like to thank our chief provocateurs and panelists for helping us understand how we can optimize patient registries to help accelerate drug development through shared data standards and clear use cases for real-world evidence in drug development. I'd also like to thank my colleagues at the Penn Orphan Disease Center, Samantha Charleston, Eric Marsh, and Monique Malloy. Their insight and experience has helped us build a thoughtful and provocative discussion over the last month, creating a very real community out of our weekly virtual discussions. Today's capstone conversation, From Brooklyn to Beijing, Global Rare Disease Registries as Global Learning Platforms, starts with a fireside chat between Dr. Amy Abernethy, Principal Deputy Commissioner at the US FDA, and Steve Usdin, editor of BioCentury. Dr. Abernethy has been a longtime proponent of building a learning healthcare system in the US to help advance both drug development and patient care. And from my own work with her at FDA, I know that she approaches both topics with both enormous insight and passion. Steve Usdin, our moderator today, is the senior Washington editor and head of policy regulation at BioCentury, where he has worked since 1993. Steve's work covering political and policy issues affecting the life sciences sector is a must-read for anyone who wants to understand how regulations, technology, and yes, personnel impact the industry's ability to translate advances in basic science into safe and effective FDA-approved drugs and biologics. Steve's reporting on biotechnology and biomedical policy has been cited in The Economist, The Wall Street Journal, The Washington Post, The New Scientist, and many other publications. In 2012, the FDA Alumni Association named Steve the Harvey W. Wiley Lecturer, making him the first journalist to receive the Wiley Award. Steve doesn't just follow the industry, he uses his deep insight into how it works, or doesn't, to challenge received wisdom and think seriously about the future of biotech innovation and regulation. After Steve's interview with Dr. Abernethy, he will pivot to a panel discussion with Elizabeth Powers, Vice President and Category Lead for Safety Evidence for Regulators and Real-World Solutions at IQVIA. Elizabeth's responsibilities include applying advances in data and technology to post-marketing requirements with the aim of driving greater value for evidence generation. Elizabeth will be joined by Jill Weimer, my colleague and Senior Vice President for Discovery Science at Amicus Therapeutics, as well as the Director of Therapeutic Development at Sanford Research, where her research program focuses on the molecular mechanisms mediating development of the cerebral cortex and how disruption in these processes can lead to a whole host of neuropediatric disorders, including Batten's disease. Work in Dr. Weimer's lab helped lead to the first ever gene therapy programs for CLN3 and CLNL6 Batten disease. They'll be joined by Sean Cozen, who is currently the Global Head of Data Strategy at Janssen, and a former regulator at the FDA's Oncology Center of Excellence, where he led the center's bioinformatics efforts and was the founding executive director of the Information Exchange and Data Transformation Lab, Informed. This was US FDA's first data science and technology incubator developed under special authorities from the US Department of Health and Human Services. Finally, we'll be joined by Glenn DeVry, co-founder and co-CEO of Metadata, the most used platform for clinical trials around the world. Metadata has powered tens of thousands of clinical trials with millions of patients and billions of patient records, advancing pharmaceutical business transformation with technology and non-traditional ways of thinking, leading to greater industry collaboration. Following the panel discussion, Eric Marsh, Associate Professor of Neurology at the University of Pennsylvania and the Children's Hospital of Philadelphia, and Clinical Director at the Penn Orphan Disease Center, will offer some closing reflections on what we've learned over the last month. With that, I'd like to hand the conversation over to Steve Usdin.
Thanks. It's really a pleasure to interview Dr. Amy Abernethy. Dr. Abernethy is Principal Deputy Commissioner and Acting CIO at the FDA. Prior to joining FDA, she was Chief Medical Officer and Chief Science Officer at Flatiron Health. She's dedicated her career to finding ways to ensure that the experience of everyone with a disease or condition benefits those who follow them, and that each individual benefits from those who have come before. For the last 6 months, Dr. Abernethy has been leading critical aspects of FDA's response to COVID-19. The speed and intensity of that effort is extraordinary. Although it's largely invisible to the public, the changes throughout the biomedical ecosystem are real and profound. I've spoken over the last month with senior officials at FDA, CEOs of large and small biopharma companies, the leadership of Operation Warp Speed, and patient groups. They all say the same thing, that we should never go back to doing medical product development and regulation as it was done before the pandemic. Vaccine and drug development programs that ordinarily would have taken a decade or more are being compressed into a year or less. Barriers that held up the collection, sharing, and analysis of real-world data and its transformation into real-world evidence are being overcome. All of this is happening because the whole world is experiencing a hint of the anxiety that patients and families with rare diseases experience every single day. This level of urgency must be carried over into every other area where there are serious unmet medical needs. If a vaccine can be created from nothing more than the genetic sequence of a coronavirus in a year, patients with genetically defined rare diseases shouldn't have to wait decades for treatments. So I want to start a conversation today by asking Dr. Abernethy about how FDA and the healthcare ecosystem have mobilized to respond to COVID-19, and then talk about how the lessons from this experience can be applied to rare diseases. COVID-19 is an extreme example of the need to gather and analyze real-world data in real time. We started off knowing nothing about the disease or how to treat it. Amy, how has the FDA approached the task of collecting data, making sense of them, and disseminating them?
Steve, it's an honor to be here with you today. And your preamble, and the point that you made about the urgency that patients with rare diseases feel and the urgency being applied to COVID-19 is really spot on. And let's talk now about what we've been doing with respect to real-world data. If I think back to March and the sense of, just dire need to understand what was going on. Actually, in September, the dire need to understand what's going on. And looking around and saying, well, there are certainly clinical trials getting done, but there's just no way that clinical trials are going to be able to answer every single question sitting in front of us, and not with the pace that's needed to start to figure out the natural history of disease or what to do. We asked the question, how can we start to use other data sources, electronic health record data, claims data, information directly from patients, such as patient-reported outcomes or information from biosensors, or even social listening data, to try and understand the pandemic better and develop a sense of the pathway towards therapeutics, diagnostics, and vaccines. So in March, we asked the question, who are the actors who aren't yet at the table who might be able to help with this story? And that includes health tech companies who have data and have been curating them in a variety of ways. It includes health systems who may not necessarily be engaged. It includes academics who might be ready to work on COVID-19 as a problem, but aren't currently equipped with the access to the right data sets or the questions to ask. And in partnership with Reagan-Udall Foundation and Friends of Cancer Research, we started an activity called the COVID-19 Evidence Accelerator. It's managed by Reagan-Udall Foundation, and that's the foundation for FDA. And FDA then sits as an active participant in the Evidence Accelerator, as well as a technical expert providing technical expertise. And within the context of this Evidence Accelerator, it gives us a mechanism to bring all these different actors to the table to start to learn how we're going to put real-world data to use in service of COVID-19. In the next few minutes, I anticipate we'll talk about some of the tools that have been developed and what we've been doing. But the first part of this was to actually say who all needs to be activated and what kind of data sets could help us start to address the questions of COVID-19. And that's what the Evidence Accelerator has helped us do.
So I also want to remind the audience before we go on too far, I'm going to ask the audience for questions. So if you have questions, I think there's a chat button or something like that you can, Q&A button that you can hit and type the questions in. And when we get to the right time, I will ask as many of them as we have time for. So Amy, the last time we spoke about the Evidence Accelerator, you said that one of its purposes is to help join up the data pipes, I think is the expression that you used. And the idea is that there are all these actors that you said that came to the table, but they have incompatible systems, even with the best of intentions. They can't just start connecting right away. Can you describe a little bit what that problem was and how you've dealt with it?
Sure. So first of all, if I think about the different kinds of questions, and let me just hit on that, and then I'll come to the data pipes. So some of the questions in COVID-19 is, what does the disease look like, and essentially the natural history. Other questions include, what treatments are patients receiving? Are there any unexpected adverse effects? And what can we understand about treatment impact in terms of whether or not treatments might have a signal that they are successful treatments for COVID-19. Other questions are about the diagnostic tests that we're using for COVID-19, such as RT-PCR tests or serology tests, and how are those tests performing? So depending on the question to be answered, sometimes the data sets are ready to go, and other times we actually need to start to put the data sets together first. And specifically within the space of diagnostics, one of the challenges has been that the information about the manufacturer of the test is separate from the information about the result of a test for a particular patient, is separate yet again from the information about how that person fares across time within the context of having COVID-19. And so before we could even start answering questions about the diagnostic tests themselves, we had to figure out, how do you marry up all of those different types of data sets? Specifically, I always call this connecting the pipes, so that the information is ready to be analyzed in one connected data set. And this is an example of the kinds of problems we've had to solve if we're going to bring real-world data to the table to start to address the critical questions in the context of COVID-19.
So those problems are not new, and they're not unique to COVID-19. So I want to kind of segue now to the rare disease experience, because those are exactly the same problems that anybody is going to have to deal with rare diseases, except you could argue that in rare diseases, it's even more urgent because real-world data is so important, so critical for making progress against rare diseases when you've got, in many cases, very tiny populations, and you've got lots of registries, you've got lots of disconnects between the different aspects of the healthcare ecosystem. So what are your thoughts? First, the things that you're doing for COVID-19, the evidence accelerator, the other things that FDA is doing, are they going to persist, and are they going to be applicable? Are you creating infrastructure now that 6 months or a year from now will be applicable for the rare disease space?
So let me take this in two parts. First is around the infrastructure, the capabilities we're creating right now through the evidence accelerator that it's applicable to the rare disease space. And the answer is a huge resounding yes, that this is going to be highly applicable. And let me give you some examples. Part of the way that we're getting our work done in the evidence accelerator is building a set of common tools that allow data holders, analysts, researchers to basically be able to do their work more efficiently. This includes, for example, lists of common data elements we can all agree on, lists of research questions that we understand in a common way so that we build data sets to try and address them, common protocols so we have very efficient mechanisms to agree on what should the analytic methods look like, and then also places to come together, essentially virtual forums where people can learn from each other really quickly in a collaborative space so that we don't have to do the rework. And we've already talked about the work that we're doing in connecting up the pipes, which is actually showing what does it look like when different data sets are connected together so that we can solve for data gaps and we can create bigger data sets. And so those are the kinds of things that are happening within the context of the evidence accelerator. They're transferable to the rare disease space, and I hope to see them in the rare disease space. I would say that there's another thing happening in the evidence accelerator that may be as important, if not more important, which is building familiarity among FDA and among all of the actors who are coming to the table in the context of the evidence accelerator around what is possible, which is really helpful because then people can start to imagine, aha, here's how I can use that in my work. Now, when I think about the rare disease space, so going to the second part of the question, there are a couple of additional problems to solve for. So as you pointed out, the populations are small. A person might be in Pittsburgh, the next person might be in Oregon. But it's not just the populations are small. Oftentimes when you've got a rare disease, you don't just go to the same place all the time to get all of your health care. You might go to Philadelphia for part of your care and then need to go to San Diego for something else. You might actually live most of your life in Florida. And so the ability to connect up all parts of your story is often more complex in the rare disease space. And so within the context of the Evidence Accelerator, some of the work that we're doing can help solve for some of those complexities that show up in the data organization within which we see in the rare disease space. And I certainly hope that we will basically start to use these same kind of tools to be more efficient in rare diseases.
So one of the things that's been kind of a theme through this whole virtual meeting is, well, first is a frustration. People see where things ought to get, and they see where we are now, and there's a frustration that we haven't gotten there. And then I think that people, there's kind of two different schools. It's oversimplifying, but there are two different approaches to that. Some people think that really it's just time. FDA should just mandate standards and rules and practices and force the companies to do the right thing. And there are other people who argue against that and are more in favor of a consensus, what I would call a bottom-up approach. So on that continuum, where do you stand and why?
So, I stand sort of in the middle, and why? I think that's the most important part of the answer. Practically speaking, we're still developing the methods. We're still developing the methods as a research community. We're still understanding those methods and what's reliable for different problems as the FDA and as regulators. Data sets are every data set is different and has its own features, characteristics. I always say every data set has warts and every data set has opportunities. And so the need to understand data sets. And so as a community coming together, we need to be able to have the opportunity to allow innovation to happen in the context of the bottoms up. And I worry that if there's too many rules set up right now, then that's not going to allow the flexibility of developing all the different innovations that are possible within the context of the real-world data space. Innovations around generating data sets and linking data sets, and innovations around characterizing data sets or even making them better. Innovations around new analytics, innovations around what use cases or tasks we can put real-world data to work in. And then I also believe that FDA does need to provide directionality, be able to help everybody understand, here's what we're thinking good looks like. Whether that is standard ways of characterizing data, which I see as an important way of helping to differentiate different data sets and different tasks that they can be used for. Helping to define high quality methods or analytic methods, as well as to signal here are risks that that are easy, or sort of traps that are easy to fall into in this kind of research. Then also getting clear about what use cases we can put real world data to task and where we need to take caution. And so I think we need both sides of that equation in order to take this space forward. At the end of the day, this is still scientific development.
So one of the other things that I think has been a call for in this in this virtual conference has been for FDA to provide more insight into so people can know upfront what they need to do to have data that's regulatory grade. They can be used for making regulatory decisions. It sounds like you're at least endorsing that idea, the idea that FDA should provide more guidance or advice to organizations that are creating registries, so that they can know upfront what they need to do to ensure that the data they collect is going to be valid for the purposes that they're trying to use them for or anticipate using for in the future.
Absolutely. I think, Steve, this, I kind of started off as a clinical trialist, way back in my younger days. And, I think about generating clinical trials data sets, and we have sort of a set of standards or expectations that provide clarity around what to do and make it very efficient, right? Regulatory certainty or regulatory clarity is efficient because it sort of gives you the guardrails within which to work. And practically speaking, with respect to data sets, understanding what good looks like so that if you're a data set developer, you understand where to aim is going to be very important. One of the challenges is that real-world data sets aren't clinical trials data sets. And in fact, clinical trials data sets have warts too, right? So clinical trials data sets are definitely not perfect, even though we sometimes think of them as a gold standard. That's probably an erroneous assumption. And the need to sort of define for data set developers, what are the expectations? And, I always think about the expectations around data sets and data set characterization to fall into three different kind of key categories. One is, I always sort of imagine as face validity, being able to ensure that the data set is representative of what you expect it to represent, and it sort of meets the overarching sniff test that everybody can agree to. The second thing that I think is helpful to understand about data sets is the reliability to which the different elements within the context of the data set actually represent what they're supposed to represent. And then the third is validity, to the ability of the data set itself to ultimately provide valid evaluation of the question at hand when you put it through a series of different kinds of experiments. That's Amy speaking, not FDA speaking, but those kinds of characteristics and being able to clarify what are our expectations around the data set to hit in those sort of three characteristics will help us to understand what does good look like with respect to data. Now, again, that's Amy speaking, not FDA.
So, well, and then FDA should be listening. One of the things that I think that's changing also is the kind of the time frame that people have to think about registries and real world data with new modalities. And for example, with gene therapies, these registries they're gonna have to persist for 10, 15 years, maybe longer. What are the challenges in that, and how do you think about governance and sustainability to ensure that data is going to be collected and it's going to be useful over time periods that are different from anything I think that we've ever thought about before?
So let me first hit on time in two directions, Steve. One of the important aspects of real-world data sets is that we're now at a place where real-world data can represent up to a second ago, right? So we can have data sets that are near real time. And that has its own set of challenges as well as opportunities. So again, whenever we think about applying data sets to the right task, there are issues as it relates to the timeliness of the data and what you can do with it. And then there are other aspects of real-world data sets where it can help us solve for these long time horizon tasks. The real-world data that allows for 15 years of data collection after novel gene and cell therapies, for example. And so both tasks come into play. As we think about this issue of the longitudinality and the issues of governance, right? Things change a lot over 15 years, and capabilities change. There's going to be changes in technical capabilities, changes in our understanding of data. People come and go, and social expectations come and go. And so being able to have upfront a general plan for how we're going to accommodate for those changes, but also an expectation of here's what we're trying to get to. We're trying to understand across time safety signals. We're trying to understand across time continued efficacy. And being able to maintain clarity around what it is that we're trying to do with this particular data set is a really important part of the overall governance plan, including a governance plan that sort of says here's how we're going to accommodate for change. The other thing that I would say is that ultimately being able to weave in new capabilities. And one example that I'll just give is tokenization. where, that's sort of a capability that it's been around for a while, but might be relevant to start applying to some of our real world data sets now, which allows essentially to give a individual kind of numerical signal attached to an individual person that will allow data sets that are generated about that person to now be pulled into a common place across time with a lot less work on that person's behalf or by that person in order to generate those kind of data sets. And so being able to have forethought about applying new capabilities like tokenization to support long-standing data sets that need to be generated over years is the kind of thing that I think we need to think about weaving into what we do. The last thing I would say is I hope to continuously see innovation in this space. And so as innovation happens, being able to have models that incorporate new innovation is going to be.
So I want to shift over to the questions from the audience, which are actually some of them are the same I was going to ask anyway. So it's great. One of the questions from the audience is, how can patient advocacy groups best assist in this process or get involved, collecting, curating data, supporting initiatives, etc.? And I would take it a step further. I mean, my bias is that that as much as possible should be directed by the patient groups. Patient groups are the ones who have the most stake here, and they have the most knowledge and the most passion for obviously for collecting this data and for ensuring that it's used properly. So I guess the question is, beyond first, how can they best assist, but also what can and should FDA do to empower patient groups?
So, your point is spot on, which is patient groups have really the most stake here. And that includes ensuring that data are used as intended and data aren't hoarded. And importantly, patient groups can help to ensure that we've got the right conversations happening related to privacy, around making sure that when people leave their legacy through data, we honor and adhere to that legacy by making sure that the information is used as intended, but that the information gets used. One of the things that always worries me is when information doesn't get used, because we're either hoarding it or we forget about the fact that it has the opportunity to contribute. Patient groups can also think about how to ensure people have the opportunity to participate if they want to. So, and also that the data sets represent what really matters. One of the things that I'm sort of struck by often is that the data sets that are intended to be collected are disconnected to the experiences real people have with the illness, and we don't incorporate those experiences as really critical endpoints or outcome measures within data sets. And patient groups, I think, are really in a unique position of ensuring data sets are representative to what matters. You asked, how can FDA ensure patient groups are represented? And I think our Oncology Center for Excellence really provides an important roadmap here. The Oncology Center for Excellence interfaces with many different patient groups, and that ensures that not only is the patient or sort of the person voice heard, but also ensures that the patient groups knows what matters to FDA, so that there is some lockstep that's happening there, which is very important. Another thing that I've seen both in the rare disease space as well as in the oncology space is for FDA to have listening sessions with patient groups, with real people and families experiencing the illness, to hear what matters in day-to-day life. And an example there that really struck me, I was in a listening session with families of children who have neurodegenerative diseases, and really understanding the impact on sleep really struck me as an important outcome that is often missed. And so being able to facilitate that kind of key understanding is really important here. And then the last thing I would say is patient groups should work with FDA to make sure that patients are fully represented in the work that we're doing.
Thanks. Another question from the audience. Someone asked, do you see the future of real-world data as intentional, that is, as in placebo arm, registry, natural history data sets, unintentional, unfocused, claims data, federated EMR data, or a combination of the two?
Combination of the two. And, so when this sort of idea around intentional data collection, so information being pre-specified and collected on some type of predefined cadence, that kind of information is really important because usually it ensures higher quality data points, and it makes sure that we have a unified enough cadence that we don't miss information that's important that needs to come in. That should be married with information that's more passively collected and may be able to better represent the totality of a personal experience. So information that lives in claims, electronic health records, lives on your watch. That is information that really provides kind of a more complete story. And I don't see this as either or. Both of those types of data sets, the intentional and the passive, have different contributions to the overall story.
So here's a question from the audience. I think this is a question for Amy rather than for the FDA. Is a rare disease center of excellence what we need, something like the Oncology Center of Excellence, to organize the movement and needs for rare diseases?
So I think that looking at the Oncology Center for Excellence model is an important thing to do regardless. Whether there is a formal center or not, understanding what's working about the Oncology Center for Excellence model and also the things that don't work will help us all understand how to accelerate the path towards, making sure that there are more and affordable solutions to rare diseases. And examples of what's working. So first of all, the ability to do novel and innovative regulatory work. So, for example, real-time oncology review. There's not any reason why that needs an O in the story. It really should be real-time review. And so, figuring out how do we move beyond the oncology space for that is an example. Another example of seeing what works is the ability within the Oncology Center for Excellence for the reviewers to learn from each other and learn from the community in a really fast-paced, innovative way, because that actually helps to generate new ideas about how this work gets done. And that's partly because there's also a lot of forward thinking around research going on within the Oncology Center for Excellence, which again builds this familiarity with the possible within the Oncology Center for Excellence, Oncology Center for Excellence, and among their staff. And that familiarity with the possible then gets embedded in how we think about regulatory science, but also regulatory capabilities and action. And that, I think, pushes the whole space forward. So that kind of thinking we need to see in rare diseases. Again, Amy speaking, I'm not speaking for the FDA, but I have watched the Oncology Center for Excellence and found it quite remarkable to see how those things move forward.
Another question from the audience. Someone asks if there are examples of rare CNS diseases or disorders that have benefited from these kinds of initiatives in I guess the question is referring to real world data, real world evidence. And I'll marry that up with another question, which was somebody asked if there are any examples, and they asked specifically also about CNS or neurological disorders, but it could be more general. If there are examples of natural history studies or natural history data that's been used as control arm in the rare disease space.
So I can't give you the exact diseases off the top of my head right this second, but yes, indeed, there are a number of case examples where natural history data has been used as essentially the benchmark or the comparator. And this makes sense because many times in the rare disease space, there are so few people with the illness that really we need that historical benchmark to understand how to think about any new therapy. And so I'm an oncologist by background, and so I tend to have an oncology frame of reference, and so I can't really kind of describe that for the CNS space specifically. But yes, that has been an approach used within the context of the rare disease space.
So somebody asked, and this is another, I think one of the themes that's really run through this whole virtual meeting, which is, should patient advocacy groups proceed with intentional real-world data collection efforts while they wait for FDA guidance on regulatory grade data, or do they risk that any data that they're collecting won't ultimately be usable? In other words, well, I think part of it's a plea for help. They want FDA to move faster, but they're also saying, you know, should they wait? Should people be waiting, or you know, how should they proceed?
So, you know, my strong bias is that people shouldn't wait, but that should be ready to morph as more understanding comes forward. And that's not just about the FDA. It's about the practical reality that even if we do provide more guidance around sort of concepts of regulatory grade data, or over, you know, as the person who managed to write a number of papers around this topic, I now sort of reflect that it's a term that's sort of somewhat loaded. But you know, the reason why I think that really we shouldn't wait is because there's going to be continuous evolution in expectation and capabilities, but there's always the opportunity to learn from what we're already doing right now, even if it needs to evolve. So the most important part is to have a governance model, to have a data collection approach, to have elasticity in thinking that accommodates the fact that things are going to continue to change across time. And you know, the other thing that I would say, and this is something that we're, you know, talking about right now a lot in the Evidence Accelerator, is that just because a data set can't answer a question about a drug approval sitting in front of the FDA doesn't mean that the data set can't answer important questions. Many of the important questions sitting in front of us, and we know this from COVID-19, is about describing the illness, about understanding the needs, figuring out patterns of care, understanding what quality of care looks like. And those kinds of questions are important and don't require some of the specifications that are likely going to be required of data when the data needs to come to FDA and be cross-checked to make sure that each data point represents exactly what we expect it to represent. So the importance of data that can help describe, the importance of data that can allow us to evaluate performance across time, including you know, quality of healthcare delivery, those data sets can be collected right now and should be collected right now, and are as important to figuring out what's going on as anything.
Well, you know, that's interesting because that fits right in with an observation that someone from the audience just sent in, where they said that many of the endpoints that are used in clinical trials now don't capture what's meaningful in particular diseases. And so I wonder, you know, is one of the things that should be thought about is not how to recapitulate and do a better job or a more comprehensive job with real world data of getting the same kinds of data that's collected in clinical trials, but instead expanding out and getting data that's more relevant to patients and to the people who are trying to develop therapies.
100%. And you know, the other thing that strikes me, Steve, and just sitting here in this conversation, is that I don't know why the evidence accelerator model needs to be limited to COVID-19. Just like the question that came in around the Oncology Center for Excellence and, you know, making sure that the principles and the way that things are getting done is transmitted to the rare disease community. I think that the ways that we're moving things forward with urgency in the real world data space for COVID-19 should be also transmitted to the rare disease space, because it'll help people understand capabilities. It'll help people build off of each other's ideas and do so with sort of an efficiency and speed that we need in order to take better care of each other in the rare disease space.
Well, I think one of the things that's really fascinating about the evidence accelerators, people who are in the audience who are watching haven't seen them, they should log on and find them. There are these weekly meetings that are held, and then there are notes that are posted that are public about it. The transparency of the whole endeavor is fascinating and inspiring. And I think that's really a model also.
Thank you.
And we've run out of time. I could talk to you all day and ask you questions, and I hope I get a chance to soon. But I think we need to switch over to our panel now. Paul?
Thanks for your time.
Thank you.
Terrific. And now we're switching over to the panel discussion with Elizabeth Powers, Sean Cozen, Glenn DeVry, and Jill Weimer.
Okay, so am I on? We started? Good. Okay. So the name of this session comes from the Margaret Mead quote, "Never doubt that a small group of thoughtful, committed citizens can change the world. Indeed, it's the only thing that ever has." And so I'm assuming that by asking if we're such a group, that it's a rhetorical question, that of course we are all committed to changing the world for the better. Why else would we be doing this? So I'm going to ask each of the panelists a kind of a framing question to elicit their ideas about how to bring about that change, and then ask them to spend 4 or 5 minutes talking about it, and then we'll have a conversation around the things that come up. I want to start with Elizabeth Powers. There seems to be a broad agreement on what needs to be accomplished, where the rare disease community needs to go, but there are different ideas about how to get there. And there's a frustration over a lack of standards and failure of parties to collaborate and share in the best interests of patients. And this has led, as I mentioned to Dr. Abernathy, to demands for top-down government mandates. There's also a view that that stepping in and creating standards and dictating them would cause more problems than it would solve. Where do you stand in this kind of continuum?
I guess as I've listened to the discussions over the last month, I've had 3 kind of strong reactions. One is I do feel that one has to beware of overstandardization. That what happens, we see this time and again in our work with rare diseases, that the data needs are different, and one standard format doesn't actually meet the needs of different diseases. And we also see time and again that actually taking a TA by TA approach actually leads, you can achieve clinically rich data in a standard format, but you really need to look deeply at the TA and the needs of the patients and the needs of the different stakeholders. So I'd say that's one thing, and that trying to achieve standardization across diseases is probably an exercise only to achieve less standardization in the end. So that's one thing. I'd say a second thing is that, and Amy Abernathy just kind of referred to some of this. She talked about, the importance of not data hoarding. And I think there really does need to, and I don't think anyone kind of intentionally data hoards. I don't know. But I do think adopting an intent to share is really important. And starting with that as a mindset and thinking about that as you think about consent and privacy. And I think we're seeing some interesting plays there, like the World Federation of Hemophilia and a couple of other registries beginning to really work through that. And then if I think about, okay, so I've said I think overstandardization is a risk. What do I think someone like a group like the FDA, an agency like the FDA can do? And I think part of what they can do, and it gets into this learning healthcare system. I'm seeing so much influence from the FDA come about by their experiments. So the work that they're doing on Sentinel and rapid analytics, people are experiencing that and wanting their own. And I think that those types of experiments have incredible impact. So let me just pause there. Those would be kind of the 3 big things that I would say in terms of response to okay, we need a mandate that says we're going to standardize, and a kind of middle of the road position around that.
So I want to switch over to Jill and ask you to describe the coordination of rare diseases at Sanford CORDS and talk about what lessons you think might be learned from that approach that might be broadly applicable to the discussion that we've had today and in the previous couple of 3 sessions.
Absolutely. So Paul mentioned in my intro that I sort of have 2 full-time jobs working with the Amicus team to oversee their science division, but I also oversee therapeutic development at Sanford Research, which is a large rural healthcare network positioned in the Northern Plains. And I have an academic research lab there. And one of the programs that probably about 10 years ago, owing to a call from the rare disease community that we set up, was this CORDS registry program. We saw that a lot of rare disease teams were investing a lot of time and energy in developing non-standardized, almost Excel-based registries. And so what we did as an organization is invest money to partner with these foundations to set up rare disease specific registries in a more standardized fashion, free of charge to the partnering foundations. Over that time, we've grown to 70 different disease registries, having 70 different foundation partners. We also, a person coming into the registry, if there's not a partnering foundation for their specific disease, where they would get a disease-specific questionnaire, they can also enter into a general questionnaire so that folks that are still looking for a disease area that we don't have a specific topic can still pull general data on those specific diseases. So over the last probably 3 or 4 years, we've started to take a deep look at how that data is used and what we can do as more of an academic partner to really kind of get the data out there and make it fit for purpose. And I think that Amy really resonated something that's come up over the last month in this workshop is what is fit for purpose? How are these registries being used? And I think over the last month, we've talked a lot about sort of how the agency might view the data that's coming out of them and might be useful for IND filings or different regulatory documents. But we need to remember that these registries are used by patient advocacy teams, they're used by scientists and clinicians, and there's a number of different ways that they are utilized. So the other thing that we've kind of done is say, what are the things that we can augment a registry with to help investigators, to help pharma industry? So 2 of the things that we're really focused on over the next year is one, paralleling our registry with a biorepository. So how do we actually mirror the information that's kept in the patient registries with biofluid samples, cell lines, so that investigators, clinicians, pharma partners can start to ask questions about phenotype-genotype relationship. And then the other thing that we're doing is partnering the CORDS registry with a core that we have at Sanford called the Data Exchange. And this is a process for getting linked data, EMR data with registry information. So we'll be putting an honest broker in place that will be able to either within the Sanford Health Network or using tools like OMOP and I2B to be able to extract data from other site EMRs that kind of parallel. And you can envision that there's questions that an investor or a team might have that are really geared towards specific patient registries, but there are also questions that investigators or pharma might want to ask that just use a general EMR data set to mine for new patients, to mine for diagnostic codes that led to a particular genetic diagnosis, things like that. So I think that really well, I guess as the question to Amy was, while the FDA figures out what the hard and fast rules are that they want around data, I think we can continue as active scientists and active folks working in the rare disease space. There's a lot of things that we can be doing to help enhance registries and make them fit for purpose, but keeping an eye on that there are many different purposes for how these might be used. So I will stop there. Stephen, hand it back to you.
Thanks. So I think that's actually a good segue to the question that I wanted to ask Lynn, which was, I know from our conversation before this meeting, preparing for it, that you've thought a lot about using machine learning to find data, relevant data in unexpected places, and about sharing that data. So I want to ask you to talk about that and and its relevance for the rare disease space?
Sure. So I think by definition, the data that you're looking for rare disease is rare, at least rarer than other data. And I want to be very careful. I completely agree with Elizabeth around the TA by TA approach. So I'm about to say something that might sound contrary to that, but it is not. But I do think that we need to, because of that rarity, look in interesting places. And we do have the ability to aggregate data across TAs in a way that I think, unfortunately, our industry, whether you're looking in clinical trial data or real world data, seems to have forgotten is possible. And I think it's really unfortunate. I will speak, I told you, Steve, like to people in a therapeutic area that I know nothing about, and I usually tell them immediately I don't know anything about it. But I can always make an interesting example of how they think about data, almost always, which is that if they are trying to create a disease model, however sophisticated it is, for their rare disease, their oncology indication, whatever it is, if you only look in data, in research projects and registries related to that disease, you never have time point zero. You are destined to not create a good model. So if you really want to create an interesting diagnostic model, disease progression model, and you're in oncology, you better be looking in patients who you're getting data through cardiology studies or endocrinology studies. At least that's in the clinical trial world, which admittedly I spend most of my time in how I think about stuff. And I think this is ultra applicable. I'll give a COVID example and then maybe an actual rare disease example. So wouldn't it have been amazing if we were able to take the concomitant medications and adverse events tables, which, yeah, they're different in different clinical trials and registries run by different companies and different organizations, but they're all coded to the same dictionary. They all have some of the same columns, and linked it to the geographic location of patients who are in research projects. There are literally millions of them around the world, covering basically every civilized country that we were all reading about as the pandemic spread. We should have been able to prospectively, and I know this is easy to say retrospectively, but we would have been able to not just watch the marching of the pandemic across various parts of our planet, but we would have had a list of all the other comorbidities that patients who were coming down with COVID had, because oncology patients and cardiology patients and endocrinology patients and rare disease patients were getting it. We would have had a list of all the concomitant medications they were taking. We would have, if we extended our analysis to other cross-TA possible data, we could have seen their medical history. And I think today we still don't really know what the right inputs are for a good predictive algorithm for who's going to come down with a severe case of COVID-19 are. And I think if we think differently about, again, what rocks have we not turned over, sometimes some of them obvious, we can get to that data. And because, again, let's get to the topic at hand. We worked with, and I know it's a topic in this conference, with the Castleman Disease Collaborative Network. And you don't need billions of records to do stuff like this. I mean, with fewer than 100 patients, we were able to combine data from multiple studies. We used some machine learning and some fancy statistics to combine data from multiple places. We also just had some people putting their hands on keyboards for things that were relatively obvious. And by stacking up fewer than 100 patients, by adding a little bit of interesting additional scientific data from a proteomic perspective, we were able to subtype some Castleman patients and go from a drug that had 25% efficacy given to any individual patient to being able to identify disease subtype and get to like almost 70% efficacy. So this idea of taking data from different studies, doing what you can to make it fit for purpose, right? I think Jill used that word. It's like a perfect way to think about it. I think it fits into some of the calls to action that Amy had as well. Like you mentioned, where can we find data and make it useful? What can it show us, even if it's not regulatory grade? I think this data stacking across silos in a fit for purpose mindset can unlock a huge amount of not just hypothesis confirmation, but I think also hypothesis generation. And that's the part that gets me excited about rare diseases. We need to do that. We need to find the people diagnosed with them when they're in a cardiology study, when they're in an oncology study or a real world data set. Does that make sense?
Yes, and it's very provocative. And I want to ask Sean a question which actually fits very well with what I wanted to ask Sean about. which is basically your thoughts about expanding the kinds of data that are routinely collected about individuals with rare diseases, and more important, how can business cases or business models be created that support the collection of that kind of data and the harmonizing of data collection protocols and data sharing? How can that be made part of a business model so that that everyone and especially companies like Janssen are motivated to make that a routine part of the way that they work?
Sure. Great question, Steve. So I think on the first point, in terms of thinking about ways where we can collect appropriate data elements at the point of routine care, I think that's what you were referring to. And Amy alluded to this, and that centers around the intent of data collection. So obviously, when one sees a patient at the point of routine care, the intent in some cases may be different than when somebody is conducting, let's say, a dose-finding study. However, if we think about this carefully, we'll realize that the intent of data collection, in especially later phase studies, is very similar to the intent of data collection when you see a patient at the point of routine care. You want to see how the patient is feeling and functioning. Are they getting better on your therapy? Other intrinsic and extrinsic factors that's influencing a patient's experience. And that's essentially what a clinical trial is. Obviously, there's randomization for obvious reasons to control confounders. But at the end of the day, the intent of data collection is the same for later phase studies. You just want to know what's happening to the patient. Is the therapy safe and effective? That's the same question that the physician is asking when they're treating a patient. What's different is nomenclature. And sometimes it's as simple as that. I'll give you a very specific example. In healthcare delivery and routine care, and things that we think about in terms of care quality metrics, hospital readmissions, hospitalizations are thought of in the context of healthcare utilization. In clinical trials, the same data elements are collected, but it's in the context of safety analysis. So a safety event that leads to hospitalization, meaning an increased healthcare utilization, is a serious adverse event that in fact has to be reported to the FDA. So the nomenclature is different. So I think something that we can do is start to harmonize nomenclature that we use at the point of care with the nomenclature that we use in clinical trials, because the variables are essentially, a lot of variables that we'll realize in later phase studies are pretty identical to the intent of data collection at the point of routine care. So I think harmonizing nomenclature would be very important. And in terms of business models, another thing that was brought up earlier was the fact that there's a lot happening right now, and I understand why some people want the FDA to provide more clarity, and others want the FDA to be somewhere in the middle. And obviously, we need to encourage innovation, and as part of that, basically thinking is really starting to think about new business models. And in order for us to be able to share data, share resources, in order to accommodate research and development in rare diseases, one way to create the incentives is think about new business models. And I think it was mentioned that, and I believe Amy said that, we're still developing the methods. So thinking about new methods that can accommodate regulatory decision-making, and think about business models around development of those methods, and the data assets can be something that would, I believe, help move the field forward. Because otherwise, we're relying on fragmented efforts. We're all very much in favor of data sharing, both from an altruistic perspective and also academically. We have been for several years involved in promoting and helping data sharing efforts like Project DataSphere, which has produced great academic output. But if we're thinking about doing advancing research and development in rare diseases, we really need to start to think about the business models that can help accommodate pulling our resources together, and not necessarily even pre-competitively, but in a manner that's conducive to advancing R&D programs based on the appropriate business models and incentives. So, so I think it's multifactorial. There, Steve, there are no simple answers to the questions that you asked. But I do want to underscore that we do need new business models, and thinking about how we can share data and share best practices and methods that we're still developing would be a great way, a great start.
So I've got some questions, but I want to first start just by giving everyone a chance to respond to what your fellow panelists have said. If it's provoked any thoughts, or also any thoughts about what Amy Abernathy said. Let's start with Elizabeth.
So a couple of things. One is I think Glenn's point about the importance of small data sets is often underestimated. You know, and in rare disease, I mean, it's very interesting because in rare disease, you only have small data sets, really. And that, you know, you can even do predictive analytics on very small data sets and get, you know, depending on what you're looking for, get very meaningful insight. So I think that's one thing. I think another thing around new business models, Sean, your comments made me think, okay, well, what are new business models? And I was trying to think of examples. And I think one of the things that I'm aware of is this desire for rapid analytics. I think that in the world of epidemiology and clinical trials, there has been an acceptance of this takes a real insight taking a lot of time. And I think with the emergence of BHE, which goes by something else now, and Aetion, and you know, companies like that who are really focused on rapid analytics of available data, it's creating a new expectation, and we're really seeing emerge quite, it's really, I think, beginning to hit a critical mass. And that is something that could really more experience with rapid analytics. That is something that could really change the scientific business, if you will. You know, how research is conducted, what the expectations are, and add a whole different aspect to use cases.
Jill?
Absolutely. So I want to go back to one comment that Elizabeth made about hoarding of data. And as much as we try not to do this, as scientists, we do. You know, we hold on to our information. I think you see that with registries. You see that with natural history studies, depending on who's developing those. One of the things that CORDS did from the very beginning was, one, ensure that the patients that are partnered with the CORDS registry own their own data. And so any agreement or request for data usage or data sharing, there's always a member of that foundation or that disease community that sits on the committee, the scientific committee that evaluates that and approves that data usage, so that there's full transparency on who and how the data is being utilized. But it is shared out free of charge. Anybody can access it simply by putting in a data request. So I think at least in that case, there are models, and we've heard about them throughout this workshop, models where people are trying to do what they can to share the data as quickly as possible, because I think if we invest the time and energy in setting up these comprehensive registries, you know, it behooves us as scientists to get this into the hands of as many individuals as we can. For Glenn's comment on the small data sets, I think, this is something that I've struggled with for many years. I think as scientists, we are often trained to look for that needle in a haystack. And I think that big data and informatics of tools, as they become more commonplace in how we do science, it's allowing us to look at the whole haystack instead of looking for that elusive needle. And I think in rare diseases, we need to continue to do that, because if not, we're going to be designing clinical trials that take 4, 5, 6 years to actually have reach a meaningful input and endpoint and readout. But if we can draw on registry and natural history data and use it in creative ways, then I think it's going to allow us to have much more expedited clinical trials and get drugs to market much faster. I do think, too, to Sean's point about how the sort of what we use as a model and how we do business is important. Earlier in this workshop, we heard from the NIH team in the RADAR program, and I think they actually set a good example for at least laying out a framework or template that folks can use for how they set up registries, how they design registries, so that we are all kind of moving towards a common theme. But I think the important thing we all have to remember is that we have to be nimble. And I think this goes back to the comment that Amy made in response to one of the questions, that as we wait for the FDA to come up with their guidance, should patient advocacy groups still be forging ahead with setting up registries? And I think that is important, but you have to set them up in a way that is nimble. We discover new things about rare diseases every single year. And so a registry has to be nimble enough that you can add questions, that you can modify your data sets as the sort of landscape of that disease and how we capture data and utilize data evolves over time. So I think to that new practice of how we do business, I think that being nimble has to stay at the forefront of that conversation.
Glenn?
So actually, maybe I can link some of those thoughts to what Sean was talking about from a business model perspective. I actually don't know who said it first. I think history will show that it was a bad analogy, but this phrase, kind of data is the new oil, I think that thinking can be linked to a lot of bad behavior. Like oil is hard to find. Nature takes a long time to make it ready for our use, and it's a limited resource on the planet. And like, not one of those things applies to data in the least. It's just like the worst analogy I think ever.
Well, and the other thing, if you give somebody some oil, then you don't have it anymore, right? And
right.
Right.
Exactly.
Yeah.
Now, and so like that leads to hoarding and things. But I guess the one thing that you need to do with oil is refine it to make it useful. And at least that part does apply to data, right? And that's where I actually think some of the ideas that we've all been talking about can link scientifically, but link to business models. It is that refinement. It is the standardization, whether it's a small set of data or a giant set of data. I think, Jill, your point about, use the haystack instead of looking for the needle is a great one. But whatever the methodology is, standardizing the data, creating methods that are fit for purpose, and actually delivering some kind of patient value, some decision-making framework that's better than what we have now for diagnostics or treatment decisions, that's the IP of the future, and it's not the possession of the data, right? And I think if we can think about models, whether it's a business model or just an innovation environment, where it's not about who's got their hands on more data, but it's about who can create the most value out of it, I think we can solve a lot of these problems together faster. I actually, I'm not a huge fan of the phrase pre-competitive, because actually I think that like putting all that data together, making it available, creates a great environment for competition that promotes innovation, right? It's like good for competition in a way I think that's positive.
Sean, what your thoughts about this? And I'll just interject. I think that this idea that the competition should be around what you do with the data rather than the data itself is a powerful idea. But Sean, your ideas, your thoughts?
Max, great comments all around. I think to Glenn's point, if we think about the hierarchy of value in health technology and data science, hardware is probably the lowest, on the list, especially in classical computing. We're not, quantum computing is different. And then software would be next, and then algorithms and methods. There's certainly a lot of IP embedded within that. And I think the opportunity and the challenge that we have here is to develop new business models around methods that are valuable and methods that mathematically make sense and also make sense from a regulatory perspective if we're talking about the use of these methods in R&D programs. And also, I just want to make a quick comment about the data volume or size. And I think for rare disease, obviously, these patients are rare. So in terms of patient lives, these data sets can be very small. But that doesn't mean that they won't be valuable as long as there are objective variables that are available in these data sets. What I mean by that, there are certain variables that are obviously more important than others. In the world of oncology, especially the first wave of sort of targeted therapies, was all about single driver mutations. And a small data set that contained, for example, a single driver mutation that in a lot of cases trumps everything else, trumps all other basically demographic and disease characteristic factors, can make a small data set highly valuable. So it's about having the right data elements, and a lot of these data elements may not be wide in terms of patient numbers, but they have to be deep in a lot of cases, meaning they have to include genomic data and including germline, which is becoming more and more important in terms of understanding how patients respond to therapies and develop and maintain disease.
So I want to wrap up. We've already gone over our time a little bit, but I think we can push it. This is really a great conversation. The charge to the committee was, are we a community? And following on that theme, one of the things that that's come up in this whole in the last month is that there are a lot of different participants who have to collaborate together to get to the kinds of ends that we're talking about. And anytime when you're in a situation like that, there's a tendency. The easiest thing to do is to look to the person who's next to you or the person who's next to them and say, well, that group or that person, they're the ones who have to change, and to think about what they have to do. And that's often valid. But what I would challenge all four of you to think about now is what is it that your organization or the sector that you're working in needs to do and can do in the short term to bring about the kind of changes that that we need. And I'll start by putting Glenn on the spot.
Sure. No, I'm happy to be put on the spot. I think if you look at, because I'm really in that software as well as that kind of data fit for purpose methods world, I think we behave in all that stuff is the new oil, right? And if we could really figure out how to, we all have companies that we need to run. We have people who need to get paid. We have salaries. And by the way, just like everything in life sciences, that feeds the next generation of algorithms and tools, et cetera. But if we can figure out ways to facilitate some of the data sharing, some of the collaboration, some of the stacking of information, I think that would really be a worthy use of our time, both from an ethical perspective and frankly from a business perspective. I put out in a book, I wrote it down for the world to see, and I think the life sciences industry blew it with COVID because we didn't do what I was describing. Like if we had just pooled our adverse events and concomitant medications, which is data that, yes, I think we know belongs to all the patients, but sits in the basements, metaphorically, of all pharmaceutical companies separately, and everybody would have had to decide to put it together. I know that's not easy, but we could have had a better data set that researchers could have been using this whole time. And so where is the role of companies like Medidata, or where are the roles of life sciences companies? Who's there? Who's in charge of constantly saying, can I contribute in a way that makes everybody better at doing their job, at being innovative, at being competitive, whatever it is? And I think we need to all think about that a lot more, myself included.
Jill?
I think that we need to continually evaluate how we do things. And I think that this workshop has really kind of resonated with me that, you can have 20 different stakeholders depending on, as we said, like how, what the intended purpose for your registry or your data set is. But I think, every team really needs to set their strategy and decide what it is that they want to be and how they want to develop their data sets, but continuously evaluate how those are being utilized. Because I think, to Glenn's point about, data is no longer or will no longer become the IP. We can generate copious amounts of data, but if people aren't utilizing it, then it has no utility. There's no value in producing that data. So I think the more that we as the folks that are building and running and utilizing even these registries come together into communities like this and say, okay, you're doing this and there's no utility. Nobody's going to use this. But if you actually changed it slightly and did this, then it would make it more useful for the community. So I think being willing to adapt and be nimble, as I said before, I think will become more and more important. Where, as Glenn said, that the data in and of itself is not necessarily the commodity, it's how it's being used and the tools that we develop to be able to interrogate it become more and more powerful.
John?
That's right. I think our new organizations, and this is something that is already happening to us to a certain extent, and certainly happening at J&J and Janssen, where I am. We have made a commitment. A lot of our new organizations are starting to make a commitment to incorporate and integrate data science and technology into the process of research and therapeutic development. This is a very nuanced undertaking, and it requires commitment and a lot of thoughtfulness, because we need to, in some cases, redraw organizational contours and start to reshape some of the fundamental ways that we've been doing research and development as an industry for a number of years. And in order to accommodate these new innovations, there are some fundamental changes that need to occur. And that requires new talent, new human capital, but also the willingness and the ability to pivot and to ask different questions during a development program as the development programs advances. Because what we can solve, the art of the possible, right now has a different flavor than it did 5-10 years ago. So we have to be able to ask different questions and know what's possible and what's not. And there is an organizational aspect to that, and there's a human capital aspect to that. The other piece is that, and I believe Glenn and others mentioned this, that I think data sharing is important, and this goes back to creating right incentives, because as individuals we're all altruistic, but as organizations, we have to advance the objectives of the organization. And in life sciences, for many of us, helping patients and bringing safe and effective therapies, that's the number one objective. Very close to that is creating shareholder value. And whether the shareholders are in the public markets or private equity and VCs for startups, that is a major theme in terms of what can and cannot be done. So I think realignment of those incentives and creating new incentives that can accommodate sharing of data. Maybe we shouldn't even call it data sharing. It's really about combined resources to advance research and development for rare diseases. So we have to be creative and think about new business constructs, because again, as individuals, we're all altruistic. As organizations, we have to advance the organization's objectives.
So, Elizabeth, I'm going to give you the last word. We've gone over. We could have kept on going for another half an hour, I think. But I'll give you the last word, and then we'll turn it over to Eric.
I think in terms of things we need to do, I agree with the other things that have been said, but really, I think one of the essential things is cross-disciplinary capabilities in the heads of specific individuals. So I'm a business person. I play an epidemiologist on TV, and I'm beginning to become more of a tech person. I think bringing those three aspects together, epi, tech, and with a business mind, I think, because that does point to where needs are, and we'll get there more efficiently if we bring that lens to it. I would say that's one of the most important things we can continue to all do.
Well, thank you. Thank you all for a really fascinating conversation. Eric or Paul, I'm not sure how we do the handoff here.
I think we'll just turn it over to Eric for closing comments.
All right, everyone. Well, thank you guys for that great session today, which was a great end to the 4-part webinar series, Let's Get Real, which I think you all would agree was a lively and frank discussion about the issues that face rare diseases when trying to put together clinical data to be the foundation for therapeutics should they arise. Before I summarize the webinar series, I would first like to thank Dr. Tracy Dixon Salazar, who set the tone of the series by telling the story of her and her daughter's misadventure with trials and data collection, and feeling like the whole process was just a box-checking exercise. I'd also like to thank all of the chief provocateurs, Drs. Lipset, Mohlberg, and Hirsch, for their deft leading of the discussion, along with all the panel members who provided their unique insights into the issues confronting real-world evidence in registries. And a special thanks for Drs. Amy Abernathy and Ustin for the chat and panel discussion today. Lastly, I would like to thank all the individuals who logged on and watched the series and participated with questions and comments to enhance our discussions, as well as the series organizers, Paul Howard, Monique Malloy, and Samantha Charleston. I'd also like to thank the sponsors of the webinar series and all the technical help from Amicus. I hope everyone has found this series as enlightening as I have. I've been involved in the NIH-funded Rett syndrome natural history study that has been collecting a host of data over the last 15 years, but without asking some of the important questions that were raised here. Was the data collected so it could be used as a natural history arm of a trial? Did it use standardized terms that could be shared? Is the data available for all to access if they're interested? Will the data drive therapeutic development? While the answers to most of these questions, unfortunately, is no, it's because they weren't asked. These questions weren't asked when the study was designed. So there's a lot of work that clinical researchers, patient groups, and industry partners need to consider when planning these type of studies. This webinar raised these critical questions around data sharing, access, consistency, rigor, and purpose that we need before creating new registries and tools and not after. These considerations are incredibly timely as new diseases, foundations, and technology platforms are emerging daily. The first session of the webinar series addressed the importance of data standards and the need for FDA NIH leadership in setting principles that need to be followed for data, and especially real-world data, to be used in a forward-looking fashion and ultimately applied to an IND, NDA, or BLA. And that was, again, touched on in today's conversation with Dr. Abernathy. There's a need to educate and inform researchers and community about common data elements, data harmonization, and combining of data sets to generate novel insights about rare diseases. Ultimately, there may be a need for Congress to clearly empower the FDA to set standards for all to follow with expert and community input. The second session reviewed some existing successful registries and programs and asked questions about collaboration amongst groups within a disorder, funding for the registries and data collection, data ownership, and best practices for registry development. The session tried to define some of the terms that are loosely used in the fields. Words such as registry, natural history study, and real-world evidence all have various meanings to different players in the field. A coalescence around uniform definitions is badly needed. Also needed are innovative funding programs to prevent the siloing that occurs when the need for funding is tied to data generation. Again, that was touched on really appropriately in this last session today. Ultimately, altering the incentives and business models that drive physician researchers, foundations, and industry to encourage pre-competitive data, even if Dr. DeVries doesn't love that term, sharing is a critical path correction that is needed for rare disease. The third session stepped back from the rare disorders to review how oncology has been successful in using natural history and registry data, put together platform trials, and use natural history comparators as an arm of a study. One of the focused issues that arose from this discussion was to need to know what the purpose of the data is before collections. And related to this, what disorders, studies, or groups can share data in a meaningful way. Oncology has had an advantage of a genome-based approach with a solid knowledge of the underlying biology and pathways that come together to cause cancer. While a great deal is known about many of the rares, the oncology level of data, like is contained in the Cancer Genome Atlas, is lacking for many orphan disorders, particularly in neurology where the gap between gene and symptoms exists and limits the ability to generate therapeutics as efficiently as oncology has over the last decade. This session underscored that to make faster progress in rare disease therapeutics, we need to understand much better the linkage between phenotype and genotype so we can begin to develop the targeted treatment approach that has transformed oncology over the past decade. Yet realizing that with 7,000 plus rare diseases, the commonalities will only apply to subcategories of diseases, and for many disorders, there may have to be a disease-specific approach. I won't summarize today's session, as we all just enjoyed it together, but in total, I think you see that it touched on many of the topics from the previous 3 sections. I also hope that you all took from the webinar series that while technology and knowledge is at the edge of therapeutic development for many diseases, there still are a number of issues that need to be resolved to ensure efficient and successful trials and therapies over the next decade. While the series did not give definitive answers, the discussion has hopefully crystallized the need for solutions, and many potential solutions were raised and discussed. We, the organizers, provocateurs, and discussants, will take these discussions and offered solutions and put them together for the community in a manuscript that will hopefully act as a resource to make these next steps possible. We will be sending all of those who registered a follow-up survey to solicit more of your thoughts and topics on these topics for incorporation into our conclusions and how we all can continue this conversation into the future. Again, thank you all for participating in the webinar series. We hope that you've enjoyed listening and contributing. If anyone has any questions or comments about the whole series, please send them in via the Penn Orphan Disease website. Again, thank you all for participating and have a nice day. All right.